Hello students, let's see the tools of recombinant DNA technology. So in the previous video we had seen how recombinant DNA is formed. So let's see the tools, the main tools that are involved. So when we talk about the tools, the first thing that we have to talk about is the enzyme, the restriction enzyme. So restriction enzyme, as I told you, it will help in cutting it. Then polymerase enzyme is also involved. You remember DNA polymerase enzyme when we were talking about the replication, DNA replication. Then ligases. So these two are the main thing, restriction enzyme and ligase enzyme. Restriction enzyme will cut it and ligase enzyme will join it. It will act as a glue. Then second tool that is involved is vector. I told you about vector, the plasmid DNA with the foreign gene that will act as a vector. Then the third thing that is needed is host organism into which you are placing this gene of interest. That is the third tool of recombinant technology. So let's see about each one by one. First, restriction enzyme. Restriction enzyme. So in 1963, two enzymes for restricting the growth of bacteriophage were isolated. There were two enzymes that were isolated, and one of them added methyl group CH4. So methyl group, one of the group added the methyl group to DNA and other cut the DNA. So this enzyme which was cutting the DNA, that was what is called the restriction enzyme, restriction endonuclease. So endonuclease, I'll tell you what it is. This is what the one which was cutting, that was called the restriction enzyme. So what was its function? They were cutting the DNA at specific sites. If this is the DNA, they are cutting the DNA at specific site. So first a restriction endonuclease that was found out was HIND2. This was the first restriction endonuclease that was isolated. HIND2. Now what about this HIND2? That is what we are talking about. Before we go to HIND2, let's remember that I told about restriction endonuclease. Restriction endonuclease, when we talk about these enzymes, nucleases, they are of two types, endonuclease and exonuclease. So what is endonuclease? Endonuclease will make cut at specific position within the DNA. Endonuclease, endo means inside, so inside the DNA, within the DNA it is making cut, whereas exonuclease will make the cut at specific at the ends, that is exonuclease. So we are talking about restriction endonuclease. So restriction endonuclease, what it will do, it will make the cut at inside, in the sequence, inside the DNA sequence. So what did I say? The first restriction endonuclease that was isolated was called HIND2. Now HIND2, it always cut the DNA molecule at a particular point by recognizing 6 base pairs. And what did it do? This specific sequence which HIND2 was recognizing, that was recognition sequence. Now I'll tell you about the sequence. Which sequence did HIND2 recognize? See, it always used to recognize a sequence which is palindrome. Like here, suppose GTC, GAC. So it's palindrome. What do you mean by palindrome? Like madam. We say madam, from here also it's madam, here also it's madam. So in from 5' to 3' and 3' to 5', it recognizes the palindromic sequence. So palindrome of this would be CAG, CTG, opposite of this. So it will recognize the palindrome sequence and it will make a cut, not at the exact center, away from the center. It will make a cut like this, and these two DNA sequence would be isolated. So you will be left like this, CAG, CT, here G, and this is the other part. So this was the other. So this part that is left, this is called the sticky end, and this is the end where the DNA and the foreign gene or the gene of interest can be attached to. So this sequence is called the recognition sequence for HindII. Now, so how these names of these restriction enzymes, restriction endonuclease I told about HindII, how they are named. We'll see that a restriction enzyme which is given in your book, EcoR1. EcoR1, see the first letter, that is for Escherichia. Co for coli, Escherichia coli. R is derived from the strain from where it is taken, and 1 indicate the order of the enzymes which were isolated from the strain of bacteria. So here HindII, that is the second order. Like that, it is the order of the enzyme from which they were identified, from there where they were isolated. So that is how the restriction enzymes are named.